Thank you, everyone, for being here and for suffering through your not break right now. So I'm going to tell you a little bit about the work that my lab has done. My lab studies gene regulation, and we want to understand how the genes interact with each other that are in this 15q11 to q13 region. But really, the big goal of all of this is to help identify potential therapeutics for your kids. And so that's kind of the spirit in which I'm going to talk today. So in full disclosure, I receive research funding from pharmaceutical companies, the ones listed here, and so I just want you to know that so you don't think I'm trying to do anything nefarious. I listened to Rachel Callender yesterday in a scientific session, and she encouraged me to ask questions. And so the first question I have for you guys is, what do you know about gene therapy? Anything? Anybody know anything about gene therapy? Have you heard of it, gene therapy? Is it a good thing? Yes. Bad thing? Maybe, right? Yeah. So gene therapy typically means putting a gene back, and that's what we've heard about, right? We've heard that you can use a virus or something to put a gene back into a child with a disorder. But there are, in addition to this gene therapy that entails putting a gene back, there are other types of genetic therapies that can reduce gene products as well. And obviously, since your kids have a duplication, reducing a gene product is a good idea. And I don't know if you know this or not, but gene therapy has been a thing since like the 1990s. It's not new, but notoriously, there was a big complication in which a young man underwent gene therapy and he died in 1999. And the effect of that was that the gene therapy field largely went silent for 10, 15 years or so. But we're back in business on those types of gene therapies, but some of these other genetic therapies don't involve viruses at all, and so don't have the exact same risks as that failed gene therapy trial did. So I want to tell you a little bit about genetic therapies and the potential for that in Dup15q syndrome. So you guys probably know this, but I think it's a good place to start anyhow. Genes that are DNA encode RNA, which are translated into proteins, right? And the RNA and the proteins are what I refer to when I talk about gene products, okay? And that's the bad stuff, right? It's either an RNA or it's a protein that your kids have too much of that we have to figure out how to reduce. And so too much or too little of a gene product can cause a disorder. And I don't know if you knew this or not, but removal of a gene product, at least in the lab, is a lot easier than putting gene products back. Because when you put a gene product back, you have to figure out how much of it you need to deliver, where you need to deliver it to, and all of these things. Whereas to knock down a gene product, to reduce a gene product that's already there, you simply have to deliver something that might cut it. And both of these can be packaged into viruses to deliver the fit therapy, and this is AAV, or adeno-associated virus, is the virus of choice now. It's a small virus that it infects a lot of people anyhow and doesn't really cause much of a disorder on its own, but it's stripped. It doesn't have any of its disease-causing, sickness-causing pieces, but instead you replace the viral genome with your gene of interest. Or perhaps an RNA cutter to reduce a gene product. But there are also non-virus therapies, as I mentioned earlier, and these are sometimes siRNAs, small interfering RNAs. And what they do is they cut the RNA, they cause the cell's natural machinery to cut its own RNA, and that reduces the RNA and therefore reduces the protein. And then more commonly now are these antisense oligonucleotides, or ASOs, that also do the same thing. They bring the cell's own machinery in, cause a cut, and that also reduces the RNA and protein. And how many of you have heard of the recent clinical trial for spinal muscular atrophy, or SMA, by Ionis, Spinraza? That's a similar, that's an antisense oligotherapy. So So I just told you that there are ways to reduce gene expression and that there are genetic therapies that are potentially doable. So which gene do we attack? And you earlier heard a talk from Dr. Ben Philpott talking about the importance of the UBE3A gene. And so if you have to zone out now or if you need more coffee, I'm going to go through some things to tell you that UBE3A is a really important gene when we've studied human cells as well. So feel free to zone out if you need to. Your kids have extra copies of maternal 15q11 to q13, and that causes the syndrome. And some of your kids have maternal interstitial duplications, and this is kind of a repeat of what Brenda Finucane said earlier this morning. Some of them have maternal interstitial duplications where you have 2 copies of an imprinted region in tandem. Some have maternal interstitial triplications, which have 3 copies in tandem, and many of your kids have this isodicentric chromosome, which means there are 2 extra copies on this little extra chromosome. And in all of these cases, these all cause DUP15Q syndrome, and the extra copies are on mom's chromosome 15. And then there are some of you in here who may even carry a paternal interstitial duplication, but you're most likely the parent of a child with DUP15Q syndrome. And in this case, it's on dad's copy, and that's why it doesn't, if it causes a disorder, it's a different disorder, and it's not really, it doesn't look strongly like DUP15Q syndrome. So if we zoom in to those little yellow boxes, you can see that there are a handful of genes. And these genes are imprinted, which means that some of them are expressed only from dad's copy. The genes in blue are made only from dad's copy. The gene in red here, UBE3A, is the only gene that's made exclusively from mom's copy. And in gray are genes that are made from both mom's and dad's copy. We think in equal amounts, or roughly so. I've drawn here the lines that represent, for instance, the span of many kids with IDIC and the span of individuals with maternal DUP15Q to kind of give you a sense of which genes are duplicated. So if the genes between these little balls are actually where the genes that are duplicated in the kids. And the breakpoints, as we talked about, breakpoint 1, breakpoint 2, 3, 4, and 5 are listed here. And that's where the duplication might begin and end for many of you. So the only difference between individuals with paternal interstitial duplications, who are largely unaffected, and the individuals with maternal duplications who have DUP15Q syndrome is really this one gene, UBE3A. And another important point for us studying this disorder is that everyone with a maternal duplication involving the region, at least between breakpoint 2 and breakpoint 3 here, has DUP15Q syndrome. It means it's fully penetrant. Everybody with that duplication has the disorder. And that's kind of important, so we can make some assumptions when we model this disorder. So, which gene products need to be reduced to alleviate the bad parts of DUP15Q syndrome? Well, my lab uses technology called induced pluripotent stem cells to study this, and you've already heard an introduction, and so this is also a repeat, but I think it's helpful to hear it again to remember it. Oops. Induced pluripotent stem cells are functionally equivalent to human embryonic stem cells. And people originally studied these because they can be grown indefinitely in the lab. We can keep them around forever. And they can become any tissue type in the human body. And whereas human ES cells, human embryonic stem cells, were generated from discarded human IVF embryos and kind of carry this ethical issue, iPS cells are made in the lab by turning on stem cell genes in skin or blood samples. And my lab prefers to use blood samples just because your kids get blood draws every now and then, and so it's kind of easy to do that and not terribly invasive. We take these blood cells, we've also done this from cord blood samples or skin punch biopsy cells. We can convert them into the stem cells, we convert the stem cells into neurons, and then use the neurons to study the disorder. I want to point out here that many of you have heard of stem cells, and I think I was asked earlier about stem cell therapies. What should we put stem cells back into my kids' brains? Absolutely not. Your kids' brains are altogether, they're pretty good. Things are in the right place. They're doing most of what they're supposed to do. And so that's not a good idea at all, and that's not why we make stem cell models for Dup15q syndrome. What we're actually after are disease models, so we can understand more about what Dup15q does. You just heard a talk from Dr. James Fink. And he told you about how he's finding what's wrong with the Dup15q neurons, and that's incredibly important to understand the disorder. But also, we think for drug discovery. It's a human disorder, and this complicated genetic duplication is really hard to model correctly in mice. So we've derived induced pluripotent stem cells from several patients with Dup15q syndrome, and this is the list that I have so far. I have IDEX interstitial triplications, an interstitial duplication, and a paternal interstitial duplication, and so these really are great tools for us to study. We carefully characterize, which really means to re-diagnose the cells with Dup15q syndrome. We look for the extra chromosomes. We'll do DNA FISH, which many of your kids may have had as a diagnosis, to show that they really do have the duplications that we talk about. We perform microarrays to determine the duplication breakpoint, and again, reconfirm that they have the duplication and nothing else. And this is really important to do to make sure that we're really studying Dup15q syndrome. We then convert them to neurons. It takes a long time, so our work is unfortunately tediously slow. It takes us 10 weeks to make a neuron that's just starting to be ready to do cellular physiology like James talked about. They take a long time to develop. We shouldn't be so surprised it takes 9 months for a human to develop, and the brain takes a while to mature, and that's really what's going on here is it's taking us a long time to get a mature neuron. But on the other hand, this is great because it looks more like human development, and that's really what we're after is a good model for human brain development. This is some data that James already talked about, and I just put it up here as a placeholder to remind me to tell you that the Dup15q neurons have physiological deficits that James has found that we can see in the lab. And he had to look at several different cell lines, and at 10 weeks for each cell line to mature into neurons, this is really hard work, and it requires a lot of manpower or womanpower, however the case may be, to do it. A lot of work, a lot of money, and tediously slow. And so, but nonetheless, we do have cellular phenotypes that we can look at, including some morphological phenotypes such as dendritic protrusions. These are like dendritic spines on the neurons. Dup15q syndrome neurons have increased dendritic spine density. Angelman have lower, for instance. And we know these about the Dup15q neurons. So with a cellular phenotype and iPS-derived neurons, we can ask the question of which gene products are increased in Dup15q neurons. And so we've done this. In our neurons, we have the whole collection of different Dup15q samples, and can see that several genes actually are increased in Dup15q syndrome. And we know the amount over typical that they're increased, and that's what the relative expression means. The 1 in this case is always set to what a typical neuron would have. So, of course, different cell lines that are exactly the same. So the last 3 in this case are IDEX. And you'll notice, for instance, here at HERC2, these last 3 bars here, one of the bars is really short, and 2 of the bars are really tall. That's variability between 2 different individuals with Dup15q syndrome. So your kids, even though they have the same exact duplication, aren't exactly alike. And you're saying, no kidding, right? You know this. But when we look at gene expression levels, that's the case too. And so there's a lot of variability in humans, in unrelated humans. And so we have to think about that. And I'll come back to this in a little bit. But this gene expression helps to inform us of which gene products to reduce in Dup15q syndrome. So, Ben talked about mouse models, and Jill talked about mouse models that increase UBE3A and why we're focused on that. So what my lab has done is, with the gene expression that we know about now, we've looked at ways to reduce that gene expression in our DUP15Q cells. So we took IDIC15 cells, and we've treated them with, first of all, this is a drug. It's a anti-tumor, antimicrobial called mithromycin. I think it's a terrible drug. It used to be FDA approved for something or other, and so it's probably not a good drug, but we had reason to believe it might reduce gene expression, and so we tested it in our DUP15Q neurons and asked, can we reduce UBE3A expression? And these are different doses. Each color is a different dose, an increasing dose of mithromycin, and we can see UBE3A come down. Unfortunately, not genes in addition to UBE3A are altered in gene expression when we treat with this drug, and so I don't think it's a very good therapeutic approach, although I'm trying to keep an open mind because a lot of drugs are a little bit dirty and have off-target effects. But I think of more interest to me are these antisense oligonucleotides that really reduce gene expression in a really targeted way. So we can design antisense oligos to, for instance, UBE3A, which we did in collaboration with Ionis Therapeutics. The red bar and the pink bar here are either untreated or treated with an antisense oligo that doesn't match any DNA sequence, so it's a dummy drug, if you will. And then the two blue cases or the light blue is an antisense oligo against UBE3A, and the dark blue is neurons from a typically developing individual. And so we can reduce UBE3A levels from those seen in individuals with IDIC15. We can reduce them to normal levels using these antisense oligos. And as I showed you in the previous slide, UBE3A is not the only gene that's increased in expression. HERK2 is increased in expression as well. We designed antisense oligos to HERK2 and showed that they can decrease it. We also made ASOs for GABA beta 3, which is another gene that can be involved in seizure disorders, and so we were particularly interested in that one. And the one gene we haven't been able to successfully develop one for yet is GABA alpha 5. So we'll go back to the drawing board and try again with this one. But the point is that we can knock down individual genes in a very targeted way using these antisense oligos. And I think that this, I should mention that Eric Levine in the scientific conference yesterday presented some really interesting data where he used this antisense oligo against UBE3A and actually rescued some of the phenotypes in DUP15Q neurons. And so I think it's really worth considering whether these would be good therapeutic approaches for DUP15Q. And I'm particularly excited about this company called Execure that I've met. They're located in Chicago, and what they do is they take the antisense oligos and they stick them to benign lipid nanoparticles and make these little hairballs, for lack of a better word, that deliver the antisense oligos to various places in the nucleus. And so in addition to delivering antisense oligos, they can actually put different antisense oligos on the same little ball. So you might envision putting ASOs against UBE3A and GABA beta 3 and HERK2 all on the same ball and reducing gene expression from several genes at once. And that might be a really cool therapeutic approach to try for DUP15Q syndrome. And so we're working with Ionis and some other companies to try to explore whether or not these are good therapeutic approaches. I do have to say, though, that remember the first slide I showed you about gene therapy. If we're not careful and we're not safe in doing this, which unfortunately translates to being slow to do this, if we're not careful, we can have a setback, right? If anything bad happens in testing this, it sets the whole drug discovery process and whatnot back many years. And so we're being careful and we're trying to make sure that this is a good idea before we talk about clinical trials or talk about pursuing it further. And thankfully mouse models and whatnot are being developed, and Jill and Ben have found phenotypes and things that we can study, and so I think that this approach will be fun. So I'm going to switch gears a little bit and talk about CRISPR. How many of you have heard of CRISPR? Is it bad or good? Good. And how have you heard of this? In terms of like a therapeutic or a tool? A tool. And so whoever said tool, that's fantastic, because I think it's a good tool. I think it's a lousy therapeutic at this point. And I'll tell you a little bit about our work with CRISPR. CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats. It doesn't matter what it stands for. The important part about it is it cuts DNA. And it can really be thought of as specific DNA scissors. And along with, so the CRISPR has two different pieces to it. The first piece are the scissors themselves, which are called Cas9. And the second piece is the guide that brings the scissors to the proper sequence. And we call these guide RNAs or gRNAs. And so what literally happens is this small RNA, a guide RNA that's shown in green here, scans the DNA and sits down at the right sequence that matches it, and then it drags the scissors. The scissors recognize where it's stuck and come there, bind, and cut. And we know exactly where it cuts relative to where the guide RNA binds. It cuts exactly 3 bases in from this PAM sequence that guides it. So why do we want to cut the DNA? Well, the reason to cut the DNA is to change or remove pieces of it. And so why would you want to do that? Well, the way I'm using it in the lab, I told you that this is a great tool. The way I'm using it in the lab is to make identical twin cell lines with and without DUP15Q. The reason we want to do this is because, remember I showed you that gene expression, those little bar graphs? And there's a lot of heterogeneity, I shouldn't use that word. There's a lot of differences between different individuals with the same exact duplication. And so how do we ever study a drug, whether a drug works or not, if 3 different individuals start with 3 different levels of UBE3A, and we don't know how well it reduces? So the solution to that is we take a cell line with, in this case, an interstitial duplication, and we try to reduce one of the copy numbers so that we've generated an identical twin cell line. So all of the background genetics and everything else are identical, except we've removed one of those copies. And so we can do this. We use CRISPRs to make one little cut, and that inserts a LOX-P site. I believe Ben Philpott showed you a picture of how he made mice, and his mice actually involved these LOX-P sites. And the LOX-P sites allow us to use an enzyme called CRE recombinase that allows us to delete all of the sequence that's between these LOX-P sites. And that's the genetic trick that we do to get rid of one copy of DUP15Q in these cells. So we've been working on this, and we were trying to be clever, and we realized that we were trying to use differences between the maternal and paternal DNA in one of our cell lines to guide Cas9 only to the maternal copy, so we can change that RNA so that it only recognizes mom's copy or dad's copy in this case, taking advantage of this little A versus G nucleotide here. And so we've tried to do this, but we hedge our bets. I'm used to being wrong. I got about a 50% success rate. So we designed 3 different CRISPRs, 2 of which will take advantage of this guide that drags Cas9 to the maternal copy, and 1 won't. 1 is just unbiased, and we have to take our chances with which copy of the chromosome we hit. And so we've done this, and we've gotten several clones to survive and go through the drug selection that we do to get the ones that have undergone the deletion. But unfortunately, we've realized that our strategy, at least with the top 2, the ones that drag it to the maternal chromosome, don't really work. And the reason we've determined that it doesn't work is you can have at least two different structures of the A's versus G's. So for instance, the dad's copy can have an A, and the 2 mom's copies could have G's, and that's what we were hoping for, because then you'd cut both mom's copies. You'd put the LOX-P sites only on mom's copy and leave dad's copy untouched. Or dad's copy and mom's copy each carry a G, but one of the other mom's copies is an A. And in this case, our LOXP sites went on a paternal copy and a maternal copy, and that was no good. And so we figured out that our cells have this structure, and so it prohibits the first two CRISPRs from working. So the third CRISPR, we're testing that now, and we have a fourth CRISPR approach. It's a little bit different, but similar to try again. And that's the reality of what we do in the lab. It fails, and we try again. But so I went through all of this, and as a therapeutic now, this type of a CRISPR approach is ridiculously inefficient, right? I just showed you how it doesn't work in cells in a lab. And so as a therapeutic for individuals, it's a terrible idea at this point. On the other hand, and the other danger of using CRISPR in this way is that one of the benefits of CRISPR is that it cuts. And once it's done cutting, it keeps cutting over and over again. And once it's killed its target, it goes and finds the next best target. So it has a lot of off-target effects. And so that's the other reason that it's not very good as a therapeutic at this point. So what about IDIC-15? So we think we can also generate identical twin stem cells with and without IDIC-15. And we've been trying to remove the IDIC chromosome from these, also using CRISPR. And this strategy was talked about in a couple of different papers towards the end of 2017 and early 2018. And the gist of these approaches is that you can design CRISPRs that either cut centromeres, cut the center part of a chromosome out, or cut repeats that are specific to your chromosome. And so we've designed CRISPRs that cut at the repeats at each of the breakpoints on the IDIC chromosome. Now, these also recognize mom's chromosome and dad's chromosome, but our strategy was, well, maybe there are more cut sites on the IDIC chromosome. And if there are more cut sites, maybe that'll cause it to be lost preferentially. And like I said, I have a 50% success rate, so the answer was not really. And we did get chromosome loss when we tried this, but we only got one chromosome lost, not two. One copy of UBE3A lost and not two. And we're looking for two copies of UBE3A lost because if we lose that extra little chromosome, then we've lost two copies and not one. And if we lost one copy, we've only lost one of the parent chromosomes. So, and let me go back and say that again where I can point to it. If we've lost one copy, we've lost either dad's copy of chromosome 15 or mom's copy of chromosome 15. And if we've lost two copies, we've lost the IDIC chromosome. And that's how we designed the screen to identify these cells. So we only lost one copy. Oops. We only lost one copy, and we lost one copy in about 4 cell lines. And so what we're trying to do now is figure out whether we've lost mom's copy or dad's copy, because even though we know that we don't really want those cell lines, it's important for us to understand whether we're cutting the maternal copy or the paternal copy. Because if we can't ever cut the maternal copy, the chances of losing that little IDIC chromosome are pretty small. But if we've lost one of each, which preliminary data suggests we've lost one of each, we've surely lost a dad's copy in one of these clones. And then in other ones, it looks like we've lost a mom's copy. So we'll keep trying and trying again until we, hopefully it's a numbers game until we get rid of that little IDIC chromosome. And so the overall goal, though, of making these identical twins is that, remember, James talked about all of the different cell lines and all of the different samples he needs to do his work and to find phenotypes. If we can make identical twins, we can find it faster. Now you only need maybe 4 cell lines, 2 IDICs and 2 identical twin controls. And that helps the process move a lot faster. It reduces the variability and helps us be certain that our therapeutic approaches are working. So in summary, we use iPS cells to study DUP15Q in live human neurons, and so we've already made iPS cells and neurons, and we've already found some cellular phenotypes and are moving right along with this process. And now that we know what human neurons with DU15Q look like and how they behave, we can start to figure out which genes are responsible for their deficits, and hopefully turn that into the identification of genetic therapies or help identify new drugs that might work well for DU15Q syndrome. We are also using CRISPR to make identical twin iPS cells with and without DU15Q to study the disorder, but the technology is really not a good therapeutic approach at this point. So with that, I'll thank the people who did the work. I work with a really talented group of people, and a lot of the work that I showed you was done by Noelle Germain and Aurélie Dumarche, as well as our Human Stem Cell Core and our Human Genome Editing Core, Chris Stoddard. And I have to thank families who donated the samples. Without you all, I wouldn't have anything to talk about, and we wouldn't be as far along as we are. My collaborators, Eric Levine and Les Lowe, and our funding sources were funded by foundations like the Angelman Syndrome Foundation, the NIH, the State of Connecticut Stem Cell Program, and pharmaceutical companies like Levo.